Good afternoon, everyone. Thank you so much for joining us today for this lunchtime program as part of our month-long March celebration of reptiles and amphibians. While we're waiting for everyone to log in and get settled, we'd love to hear from you in the chat about what your favorite reptile or amphibian is. So this can be an animal you've seen in the wild, at a zoo, a museum, it could be a pet that you have, or it could be an animal you've never seen before, but you'd love to see. So just go and hop in the chat and let us know what your favorite reptile amphibian is. My name is Skelly Hunt, and I'm joined today by the museum's research curator of herpetology, Dr. Bryan Stuart. Really excited to have Dr. Stuart here with us today. And he'll be giving us a behind-the-scenes tour of our herpetology research collections. And so any questions you have during the talk, type them in the chat whenever you have them, whenever you think of them. And I will collect them and make sure that Dr. Stuart addresses them at the end of the tour. So without any further ado, I'm going to go ahead and hand it over. Dr. Stuart.
Well, hello, everybody. Welcome. Welcome to my favorite event of the year. I'm Reptile Amphibian Day here at the North Carolina Museum of Natural Sciences. And I'm so sorry we can't be in person again this year, but I really, really, really look forward to next year where I'm optimistic things will be better and we can do this in person. So thank you for turning out today and for tolerating a virtual program. So many of you, I am the research curator of herpetology here at the North Carolina Museum of Natural Sciences. And I think when most people think of the Museum of Natural Sciences, they think of this part of the museum. And this is our downtown Raleigh campus where we have the exhibits that are open to the public and the research labs and investigative labs and so forth. And I'm hoping that most of you have been there before. If not, I very strongly encourage you to visit when you're able. But what I want to talk to you about, and this is the part that people are familiar with because it's open to the public and that's where our wonderful exhibits are and so forth. I want to tell you today about my favorite part of the museum. This fairly unremarkable looking building that you're looking at here is located out at our Gold Star Drive campus. So that's at 1671 Gold Star Drive in Raleigh, on the west side of Raleigh, where the museum also has the Prairie Ridge Eco Station, which is a wonderful place to visit, and I encourage you to do so too. And the Prairie Ridge Eco Station is open to the public and has some wonderful walking trails and informative things out there. But on that same Prairie Ridge campus is this building, and this part is closed to the public. And so I thought, well, since we have to do this virtually, why not take you behind the scenes into this building and into my favorite part of the museum, which looks like this. So that building is we call it the Research Lab. It's closed to the public. It's a specially designed building for housing fluid preserved natural history collections. So we have in that building, we have our preserved specimens of fishes, invertebrates, and of course, amphibians and reptiles, what I want to talk to you about today. And these are research collections that are used by scientists here at the museum and by scientists all over the world to further our knowledge on the natural history, biology, and conservation of these animals. And so the amphibian and reptile research collection that I'm in charge of, and I help to take care of with my colleague, Jeff Bean, who's our collection manager of herpetology, houses about 275,000 preserved amphibians and reptiles. And these collections date back to the mid-1800s, back to about 1850s. So essentially, these research collections are, I like to think of them as a natural history library, where we have this incredible amount of information in these collections on what species, what distributions of species look like in the past, and what they look like in the present. And people use these research collections for all kinds of things. So in addition to having actually the preserved specimens, we also have what's really, really important are the associated data with the questions. So are the associated data with the specimens. So for each one of these individual preserved specimens, some of which are old as the 1850s, we know where they were collected, who they were collected by, and all kinds of information such as what the habitat was like and so forth. And so if you can imagine that if we have this massive collection of 275,000 plus preserved specimens with all of those associated data, we have a really good understanding, imperfect, but still quite good, of how species were distributed here in North Carolina and adjacent areas in the southeastern US in the past. And you can imagine that so many of these preserved specimens come from places today where the species no longer occur because they're under parking lots and housing developments and shopping centers and so forth. So this is a really, really important, invaluable, and irreplaceable source of information on the amphibians and reptiles of our state and adjacent areas. And people often ask, well, why do you have so many? And one of the most common uses of our collections, beyond documenting the past and present distributions of species, is understanding geographic variation among populations and among species, so that scientists can determine what we should call one species and what we should call another. And so understanding the variation that occurs within and between populations is critically important. And so, for example, this is a series, what we're looking at here is a series of preserved specimens of Twoline salamanders from a locality near the Sandhills region of North Carolina, where based on the variation that we were seeing in these animals, we had evidence to suggest that there were two species at this locality, and in fact that the two species were hybridizing because we had individuals that were intermittent in their appearance. So that's just an example of why we might have more than one representative from a particular locality. And most of our specimens in the amphibian and reptile collection are preserved in fluid preservative. They're originally fixed usually in formalin, but then they're transferred to 70% ethyl alcohol. And that allows us to just take these specimens out of the jars and handle them and use them. We measure them, we count scales, we look inside of them to understand their reproductive condition, to look at their stomach contents, all kinds of things. But in addition to fluid preserved specimens, which make up the vast majority of our research collections, we also have dry materials. And so even though the building was designed for housing fluid preserved specimens, which require some special safety protocols in place, we also house the dried materials that are part of these collections. So for example, these are skeletons of giant salamanders, a close relative of the hellbender, which is our largest North American salamander and that we are lucky to have here in Western North Carolina. So, and as I was mentioning before, each one of our specimens has a unique number that's assigned to it, okay? Using the museum acronym NCSM for our old museum name of the North Carolina State Museum. And that individual unique number, we call that a catalog number, is what we use to permanently associate that specimen with its data, when it was collected, where it was collected, and so forth. And even though our collections go back to about the 1850s, it was in 1961 that our retired herpetology curator began a formal cataloging process in which he has started this unique tagging system. Rather than just having information on the specimens on a label that might be on or inside of the jar or tied to the specimen, this formal cataloging system was started in 1961 by Bill Palmer. And it continues today. But the big difference, and we keep that's called our catalog ledger. We keep our ledgers that have these handwritten information on when and where each specimen was collected. We keep them in these fireproof safety drawers, fireproof cabinets for safety. But today, we have a big effort going in the research collection here in the herpetology unit at the museum to digitize the data, meaning to take all of those data that are in these handwritten catalogs going back so many decades and getting them into our collection database and then portaling that data, the information, into our online database. So you and scientists and biodiversity managers and anybody who needs to know what we have in our collections can go to our museum website at naturalsciences.org and go to our research pages, and you can find our online collections, not just for herpetology, not just for amphibians and reptiles, but for all of our collections. And there you can type in a species, or you can type in a geographic area, or whatever you're interested in, and you can learn what we have for preserved voucher specimens of that species or from that geographic area, or whatever it is you type into your collection. And this here is just showing a map of spotted salamanders in our collection, some of them. So you can use them to understand what we have in our collection. And why would you want to know that? Well, people then, by understanding what we have for vouchered specimens and their associated data, they can come here to the museum to look at the specimens, to photograph them, to measure them themselves, or to borrow them. We have a very active loan program where we send specimens from our museum on temporary loan to researchers all over the world who have a legitimate need to borrow the specimens or their associated data. And then, of course, unfortunately, many amphibians and reptiles are highly sought after in the pet trade, and so will have commercial value. And of course, we want to use these and make these data available on where species occur to help them. And we wouldn't want to do anything that would inadvertently hurt them. So for our very sensitive species that have high commercial value and that somebody might maliciously use the locality information that's available through these historical records to hunt them down and exploit them for commercial purposes. For those kinds of species, we obscure the detailed information in our online database. But fortunately, for the vast majority, you're able to go in there and you can learn what we have, where it was collected, when it was collected, and so forth. So pretty great. And so what I want to do, and I wanted to tell you, a lot of people ask, well, where do these specimens come from? You know, you get the question, you go out and kill them. That's sort of a question that comes up. It's a very understandable question. And I'll tell you that today, here in North Carolina and adjacent areas here in the southeastern US, the vast majority of our specimens that come into our collection today are what we call salvage specimens. So unfortunately, amphibians and reptiles suffer really heavy mortality from vehicles. We call those road-killed specimens. And you've probably seen it yourself riding around in a car. You might have seen a dead snake or a dead frog on the road. And we spent a lot of our time in our field work doing what we call road cruising. So looking for hopefully live amphibians that are on the road that we can observe and then safely move off the road, but also to pick up those that were unfortunate enough to be hit by vehicles and are still in good condition. So the vast majority of our stuff from North Carolina comes in as salvaged specimens, primarily roadkill, but sometimes other things, such as things that drowned in nets or drowned in traps and so forth. But we do some targeted collecting where there's something that we need, either it's a county record or we don't have genetic material. I'll talk about that in just a moment. And so we will go out and collect and humanely euthanize a specimen for that purpose if there's a real need for it. And so I just wanted to tell you a couple of the stories. You know, I could just go on for weeks and weeks and tell you stories from this collection. If you could just imagine, with 275,000 plus specimens, each one of them having a unique history of where they came from and who collected them and what conditions were like when they were collected, and what that locality looks like today and so forth. I mean, we can just tell innumerable numbers of stories from this collection. But I just picked out a few favorite ones that I want to share with you. And I wanted to talk to you about this very special animal. So this is what it looks like in life. This is called a Neuse River water dog. Get to take a look at it. The Neuse River water dog is a very special salamander to those of us who live here in the Triangle or live here in North Carolina. This is a salamander that's endemic to our state. It's found only in North Carolina, and it's only found in the Neuse and Tar River drainages of North Carolina. And the Neuse River water dog was actually originally described to science in the 1920s by Clement S. Brimley, who was the founder of the North Carolina Museum of Natural Sciences. And Brimley named the species Necturus lewisi. The name lewisi means belonging to Lewis. So Lewis's salamander would be the way that you would translate the name of that scientific name. And Lewis was a fisherman here in Raleigh, North Carolina, who caught the original specimens in the Neuse River near Raleigh and brought them into the museum in the 1920s and showed them to C.S. Brimley, our museum founder, who was very interested in herpetology. And he ultimately took those specimens, described them as a new species of science, and named them after that fisherman. A fisherman named Lewis. So in any case, this is now a very special species. Not only is it only found in the Neuse and Tar River drainages of North Carolina, it's now actually, it's a species that's declined a lot over years due to water quality changes, including sedimentation. And just late last year, it was listed as a federally listed species of protection. So it's a very special salamander for us here in North Carolina, and its history began not just here in Raleigh, but here at the North Carolina Museum of Natural Sciences. So really, really interesting species with a long history. And then I wanted to tell you another salamander story, one that I was involved in not too long ago. But as I mentioned before, we have all of these specimens that document variation within populations, variation among populations, and within and among species. And some colleagues and I here at the museum and at some other institutions started going through and looking at our preserved Twoline salamanders. And what we did was this is a preserved specimen, and we went through and we digitized images of these specimens, hundreds and hundreds of them, from all different angles, so that we could go in with a computer and take accurate measurements off of these tiny, tiny preserved specimens. And ultimately what we did was based on the morphological variation, the shape, the size, the coloration, as well as the genetics, which I'll mention in just a moment. We determined, my colleagues here at the museum and I and elsewhere, that this represented a new species of science. And in December of 2020, it was named by us as the Carolina Sandhill salamander, Eurycia erinicola. And this is a species that's endemic, found only in the Sandhills region of North Carolina. It gets very close to the border with South Carolina. It may very well turn up on the South Carolina side of the border, but to date, it's known only from our state, and specifically only within the Sandhills region, which is a physiographic region, about an hour and a half or so southwest of Raleigh. And I saw somebody asked about the whiskers. Those whiskers, this is an adult male, and it's a sexually active male, meaning during its breeding season. And those, what look like two little whiskers or two little fangs, those are called cirri, C-I-R-R-I. And they're used to detect pheromones. So it's essentially for mate communication and mate recognition in these salamanders. In any case, once we were able to describe it as a new species, I wanted to tell you about our type collection, which is a collection I'm very, very proud of. This is a collection where essentially when scientists go ahead and use specimens in our collection or any other museum collection to serve as the basis for describing new species of science, those museum specimens, those preserved specimens that formed the original description of the species become what are called type specimens. So type specimens, in particular, something called the holotype specimen, are the name-bearing specimens. In other words, what the original intent of the authors were for what that species is. And it's very important to have those original reference specimens, these type specimens. When you're partitioning up a species, you need to go through and really understand, because sometimes, in a concept of a species, for example, these in these two-line salamanders, the Sandhills, Carolina Sandhill salamander was once thought to be part of a much wider geographic thing called Euryceus cerridura. Okay, and just by going through and understanding where those original cerridura specimens came from and what the original meaning of the author was when that species was described was a necessary part of going ahead and describing a new species, partitioning out the Sandhills salamander as new. So in any case, we take very, very good care of our type specimens. We house them in a great big 700 pounds concrete lined cabinet to keep them as safe as we possibly can. And the reason is that when you lose type specimens, it creates a lot of trouble for people called taxonomists, people who are identifying and naming species, because they may not be able to tell from an original publication that described the species exactly what the author was intending, or maybe some characters were not referred to. And without having those original type specimens, you can't really go back and fully understand. And so, for example, during World War II, quite a number of natural history museums in London and Germany and elsewhere were unfortunately and tragically bombed during that terrible conflict, and many type specimens were lost during that conflict. And even today, so many decades after the end of World War II, taxonomists are still trying to understand the meaning of some of these original species descriptions that were based on type specimens that were lost during that war. So in any case, I wanted to talk to you about, so here we are, we're on the looking at the preserved copperheads. Again, most of our copperheads come in as salvage specimens. Either somebody unfortunately decided to just go ahead and kill them out of fear, or especially road-killed specimens. But people like to visit our collections. We do have arranged tours, and people like to come in, and I always like to tell them, well, now you can say you've been in a room with about 30,000 snakes, and you will be telling the truth. Now, none of them are alive, but it's still a true statement. And so the next story I wanted to tell you about is one of these. And somebody just asked, what's my favorite snake? Well, I'll tell you, here in North Carolina, this would have to be one of my favorite North Carolina species. This is the eastern coral snake. The eastern coral snake it's a venomous snake, and it's very, very rare now in our state. North Carolina is at the northern end of the geographic range of eastern coral snakes. And they have historically been restricted to the southeastern part of the state, westward into the Sandhills region. And I mentioned the Sandhills, and by the way, a beautiful animal. This is what a eastern coral snake looks like in life. And in any case, the eastern coral snakes from North Carolina in our collection really serve to make an important point. And that is that in our collection, we have preserved voucher specimens of eastern coral snakes from the Sandhills region of North Carolina that were collected in the 1920s, 1930s, 1940s, all the way up until the mid-1960s. And no coral snake has been observed with certainty in the Sandhills region of North Carolina since the 1960s. And it's a very interesting question about why coral snakes seem to have become so rare or probably extirpated, meaning have disappeared from the western part of their range here in North Carolina, why they have disappeared from the Sandhills region and today are restricted only to the southeastern corner of the state. But the point I wanted to make was we're so fortunate to be able to have these preserved specimens of eastern coral snakes in our collection that were collected back in the 1920s, 1930s, 1940s. And the reason is that today we can't find that species anywhere in the Sandhills. But what we can find is this. This is a scarlet king snake, and this is a harmless snake that is a mimic of the venomous coral snake. And there's at least one other species, the scarlet snake, that is probably a mimic of eastern coral snakes as well. And if we did not have these preserved voucher specimens of coral snakes from that western part of their historical range here in North Carolina, but instead simply had written accounts of somebody saying in the 1920s or 1930s, gee, I was in the Sandhills and I saw a coral snake, but they had not preserved that specimen. Today, we would probably dismiss those records as misidentified scarlet king snakes. But because we have these preserved voucher specimens, we can go in and say with certainty, indeed, that species, the eastern coral snake, did used to occupy that part of North Carolina, even though we can't seem to find it again today. And then I've alluded to a couple of times the genetics, our genetic resources and how important they are. This is also something that we worked very hard on improving here at the North Carolina Museum of Natural Sciences, not just the preserved voucher specimen, but also high quality tissue samples that can be used by researchers here at the museum and elsewhere around the world for extracting DNA or RNA or other biomolecules for a variety of research purposes. And so today, whenever we have a specimen come in, this is a mimic glass lizard, a really, really neat legless lizard that was originally described to science in the 1980s by our former herpetology curator, Bill Palmer. It's a coastal plain species that goes from North Carolina down into Florida. And it's very, very rare. It's very hard to find these throughout its range. And recently, we acquired a specimen that had been, unfortunately, a mimic glass lizard that had been run over by a vehicle. And it looked terrible. It was even difficult to count enough scales on it to be sure that it was a mimic glass lizard. But it was, and so we went ahead and preserved it here at the museum as a specimen, given how rare and uncommonly they're encountered now in North Carolina. But very importantly, we took pieces of tissue samples from it that can be used for genetic analysis, as there are not very many molecular samples available of the rare mimic glass lizard, slender glass lizard. And we take those tissue samples and we store them in a preservative, such as RNA later or full-strength ethanol. And then within an ultra-cold freezer, this is the Herpetology unit's ultra-cold freezer, the one closest to me. And that is a freezer that we keep at minus 80 Celsius, which is about, about minus 115 degrees Fahrenheit, something like that. And we store these DNA samples at super cold temperatures in an effort to slow down any enzymatic activity that might be going on in that tissue vial, in an effort to try to preserve the quality of that DNA as long as we can into the future, so that they'll be available for science. And so anyway, this is just a peek inside of our freezer where we've got boxes and boxes and boxes of tissue samples. And might look something like this. And so here you can go into our freezer and you can find the preserved tissue sample, and it's got on it a unique NCSM number. Remember that unique catalog number we talked about before? So the body of this specimen, the preserved voucher, will be on our shelves, okay? But the tissue will be here in our ultra cold freezer. And so people can come and borrow and use not only the preserved specimen, and where they can look at morphological variation, counting scales and measuring and looking color pattern and so forth. But they can also now use a small piece of tissue to extract DNA from that very same specimen. And then I just finally wanted to wrap up by saying that although we rely very, very heavily on salvage collecting here in North Carolina as we grow our collections, we also grow our collections through explorations of poorly studied areas here in the US, but especially in tropical areas around the world, where historically people haven't studied amphibians and reptiles as well as they have, and for example, here in North Carolina. And so a big part of my research is also beyond North Carolina, getting involved in understanding amphibian and reptile biodiversity in Southeast Asia, specifically in the countries of Cambodia, Laos, Vietnam, and Thailand. And this is just a photograph from a recent, pre-COVID field trip to Cambodia with my good colleagues. The guy in the middle with the blue boots, that's Cambodia's sole herpetologist, Mr. Ning T. And on our last field trip together, before COVID, here's a picture of my tent. What we basically do is we set up a camp in the forest, and together as a team, we go out in the forest and we try to document as many different species of amphibians and reptiles as we can find. And then from some representative individuals of what we find, we go ahead and photograph them, preserve some as specimens, taking those high quality DNA samples, and then returning them to the museum, in this case, the museum in Cambodia, and some of the specimens here to the Museum of Natural Sciences for further study. And so, for example, here on that recent trip, the two of my colleagues are looking at something that's going to be very, very hard to see in the picture, but I'll blow it up here in just a second. But let's see, how do I do this? Let's see, right there, right at the end of my finger, there's sort of a dark gray worm looking thing under a log in the forest in southwestern Cambodia. And it was a cardamom caecilian. A caecilian is a limbless amphibian, okay? So you're familiar, amphibians are frogs and toads are one group, and the newts and salamanders are another group. And then the third group of living amphibians are these guys, the caecilians, which are totally limbless. They have no front or hind limbs, and they're either burrowing underground or aquatic, and they live only in tropical areas of the world. And so they're very, very, very poorly known, given that not only do they live in these tropical areas, but they're fairly inaccessible within those areas. And so this is an example of the cardamom caecilian. That's the name of this species. And this is only about the fourth specimen, fourth individual that's ever been observed. It doesn't mean that it's rare, but it's just in a fairly unexplored part of the world, and where it lives underground in that tropical forest of Cambodia. And so we've preserved that specimen and taken high quality DNA samples, and are going ahead and now getting a better understanding of how the cardamom caecilian is related to other caecilians from that part of the world. Okay. So that's all I have for prepared images, but I would love to take any questions you might have. Thank you so much.
Yeah, thank you, Dr. Stewart. We do have some questions. First of all, I had a question about the cardamom caecilian. Does it somehow interact with the cardamom plant?
Oh, interesting question. Well, it's from the Cardamom Mountains of Cambodia, and the Cardamom Mountains got their name from having the plant in those mountains.
That makes sense. They're both mapping the same thing. And someone from the chat wanted to know, how many mosquito bites did you get while you were in Cambodia last time, if you journaled that?
Many, many, many. And over the years, I've been working in Southeast Asia since I was a graduate student. I've been let's see, how many years is it now? So 24 years now I've been working in Southeast Asia, and over the years, of course, I've had lots and lots of mosquito bites. Unfortunately, I've had dengue fever and malaria a number of times, which are diseases that are transmitted by mosquitoes. So the answer is many mosquito bites.
You're probably getting immune to most tropical diseases. I guess that interacts well with my next question. What was your educational and career track that led you to the position you're in now? And how did you get involved with the museum in Cambodia?
Great questions. So I have always been interested in biodiversity and especially amphibians and reptiles. And when I was young, I didn't even know that natural history museums existed, and that you could have a job as a scientist, understanding what species occur where. Just I had no idea these opportunities existed. And so in any case, I after high school, I went to Cornell as an undergraduate and got an undergraduate degree in biology. And then after that, I came here to NC State, North Carolina State University. Did my master's degree. And on those days, I was working on snake venom biochemistry. I was also, and remained very interested in snake venom. And in those days, that's what I decided to focus on for my master's thesis. But while I was a master's student, I took a course called systematics, which is the field of understanding, basically understanding biodiversity, how species arise, how they're related to one another, and so forth. It's a study of biodiversity, essentially. And I took a systematics course over in the insect department, the entomology department at NC State. And I just thought, oh, wow, I'm really, really interested in systematics. And that was when I realized, wow, well, I always knew I was going to work on amphibians and reptiles, but I really want to be somewhere where I can do systematics and really be immersed in the biodiversity of amphibians and reptiles. So I ended up doing a PhD in a joint program at the Field Museum of Natural History and the University of Illinois in Chicago. And then I did a postdoc at the Museum of Vertebrate Zoology at the University of California, Berkeley. And from there, got this job in 2008. So, the really important thing is, if you decide you want to do research in a natural history museum, is to try to get involved in working in that natural history museum as soon as you can. And so I was lucky to, as a graduate student, to get involved and to be able to be part of the Field Museum of Natural History. And then they have a very strong program in Southeast Asia, and that led me there. And over the years, you just develop networks of colleagues, collaborators in these countries who share some of the same research interests as you, and you can really do division of labor. So, for example, my colleague Ning T, he's just a fabulous field biologist. And it just has discovered so many new species to science from his country in Cambodia. But he doesn't, for example, have the means to do DNA sequencing where he's at, just because of the costs and availability of equipment and stuff are prohibitive. And so we have often teamed over many years where I take on the DNA sequencing component, and he a lot of the field work or morphological study of the preserved specimens and so forth. So having these kinds of collaborative relationships are really, really important for working productively and ethically in foreign countries.
And on that note, what sort of opportunities does NCMNS offer for students as far as research, internships, or volunteer opportunities?
All of the above. So we do have a formal volunteer program and a formal intern program. And I would direct you to the museum website to check that out, unless anybody wants to post in the chat a direct link to that. And that's usually the best way. And so we do have those formal programs. Some are paid, many are not paid. But the best thing to do is, if you're interested in a particular group of organisms or a particular kind of research, is to find somebody at the museum or similar institution who's working on that, and contact them directly and find out, do you think there might be an opportunity to get involved in your laboratory and gain some experience for this purpose? So
And you mentioned that in Cambodia, they didn't have really robust ability to do DNA sequencing and research. Does the North Carolina Museum have more robust laboratory, or is that another connection that you've cultivated?
Great question. Thank you. I'm very fortunate to have access to a wonderful facility here at the North Carolina Museum of Natural Sciences called the Genomics and Microbiology Research Lab. And that's something you can go and see yourself. It's on the 3rd floor of the museum's Nature Research Center, which is one of our 2 buildings in downtown Raleigh. And there you can see we actually have 2 different kinds of DNA sequencers. Something called a Sanger DNA sequencer and a next generation Illumina MiSeq sequencer. And so I'm very, very lucky to have access and the ability to be able to do sequencing for my research needs, and which includes those of my collaborators, of course, here at the museum. So
I imagine it makes sense to have it close to your freezers. Really helps out with the logistics. Someone was wondering what the furthest you've ever sent a specimen to another researcher is.
Good question. Well, let's see. We've certainly sent specimens to Europe. We've sent a number of specimens to Southeast Asia. Lots of specimens to Australia. There's no limit on how far we would send a specimen. What we really take into consideration is, first, is there a legitimate need for this specimen? Is there something that we have here that person at a distant institution really can't get access to, or something they really need? So we take that into mind. And then secondly, we look at the nature of the request, what's actually being asked for, how many specimens, what are they? And by sending them off overseas or whatnot, are we likely to be able to get them safely back? In other words, are they going to go somewhere where they'll be housed in the climate-controlled conditions that we use here at the research lab? So constant temperature and humidity and protected from light and in good quality, 70% ethyl alcohol, and that they can be shipped in conforming to, there are some hazardous material, hazmat regulations that pertain to shipping alcohol preserved specimens for safety reasons. So we look at all those kinds of factors. So it isn't so much geographic distance as it is, we just want to make sure that we're the right institution to make that loan. And also, is it something that we can be sure we'll get back at the end of the loan period in safe condition, so that it's available for use by others in the community.
I imagine that there are some specimens you have that are so rare that you would never let them leave the building.
Very good question. And one of the interesting ones, we spent a little time today talking about type specimens. Remember those? In particular, the holotype, which is the name-bearing specimen when somebody describes a new species of science. Remember I showed you that cabinet? And a lot of museums are very reluctant or have policies that prohibit actually loaning out the holotype specimen just because of the risk of it being damaged or lost in transit. And so for that reason, museums will often say, if you need to examine the holotype specimen, which is really important, we need you to come and see it here at our museum.
Gotcha. So someone was wondering, what is it about 70% ethyl alcohol that allows handling, which is something you mentioned earlier, and how is that different from formalin?
Oh, good question. Well, formaldehyde. So formalin is formaldehyde solution, and formalin is really, really good for fixing the morphology. Okay, and that's why, for example, when like a little skin biopsy is taken from your doctor, and that biopsy is then transformed into a slide on a microscope, it goes through a formalin fixation. Formalin is really, really good at holding up the structure of the original cellular structure and keeping the morphology intact. Unfortunately, formalin is a carcinogen, meaning it's a cancer-inducing agent. And so we handle our formalin very, very carefully. We have a fume hood, for example, Obviously, always wear gloves and protective, we have protective eyewear and whatnot. So we want to be very careful when we handle formalin. Unfortunately, I did oversimplify when I said specimens are originally fixed in formalin and then transferred to 70% alcohol. There are some specimens that remain permanently in formalin, and those are larval amphibians, amphibian eggs, and a couple of other things like that, where they don't hold up. They don't hold up as well when they're transferred into ethyl alcohol. And so when we have our specimens that are permanently stored in formalin, we just take extra care when we open those containers to study them. Ethyl alcohol is it's of course still a dangerous chemical, but it's essentially it's not as dangerous. It's much easier to work with. We wear gloves, but we don't have to worry about carcinogen effects or anything like that. So we just remain mindful, but it's much easier to take care of. And the 70% is just that we've learned, people in the long distant past, that about that concentration is just about the right one, because you want enough ethyl alcohol to preserve the specimen, to remove enough water from it, to slow down enzymatic activity, anything like that, but not so much alcohol, like full strength alcohol, that would pull out so much water that it would dry up or change the form of the specimen. So we've just learned that 70% is a really nice for long-term storage. And of course, we hope that these specimens will be preserved for hundreds of years or into perpetuity. That's what we're aiming for.
Yeah, it does look like the copperheads behind you have retained their form quite nicely as far as the amount of moisture inside versus outside. One of our viewers noticed that the colors of a lot of these specimens appear to be muted. Is that a result of the ethyl alcohol?
Very good question. The answer is yes. Unfortunately, that's the downside of fluid preservation of amphibians and reptiles. Even though it's definitely the best way to do it, coloration does fade. And so for that reason, whenever we have something come in fresh, dead or alive, we want to try to take images of it before it's preserved so that we can retain that color information. So that is an unfortunate downside is color fading in preservative over time.
So besides the cancer-inducing formulas you work with, what are some other safety concerns you have with handling these collection items?
Well, of course, they're flammable. And these chemicals are flammable and so forth. And having such a large amount of the flammable, preservative in one place requires, we have safety alarms. We actually have ethyl alcohol alarms, meaning if there's a spill of alcohol, we'll have an alarm go off. We have special drainage in the floors. We just take great care just to make sure that nobody's exposed to large amounts of the chemicals. What I was going to say is there was a second downside to using formalin. Besides the safety issue, formalin, is wonderful as it is at preserving the morphology of a specimen. It unfortunately makes DNA extraction of that specimen later very, very difficult or impossible. So formalin really binds up the DNA, causes cross linkages, and it degrades it and so forth. And so that's the reason why when we have a fresh specimen coming in, we remove a piece of tissue of it for the freezer before it gets anywhere near to our formalin. Gotcha.
And so someone was wondering how the digitization project is going percentage-wise. I think the last time we spoke, you mentioned the ability to add photographs to the database was something coming up. How is that going?
Great questions. Yeah. So we're at, I'd have to look exact, but I'd say something like about 70%, 75% of our collection is fully digitized. We recently acquired, over the last number of recent years, we acquired the collections of two other natural history museums that decided they couldn't keep their collections anymore. And so because of that, we have an enormous amount of what we call backlog. So it isn't just digitizing the specimens that were already in our collection, but we've recently obtained so many additional ones that the databasing is a constant challenge. about the imaging, so we are hoping to move, and we're hoping it can happen this year, moving to a new database platform, a software called Specify. And Specify is a really neat database platform that is specifically designed for natural history collections. And so it gives us all kinds of flexibility, lots of really neat tools. And one of the things we want to be able to do is start attaching some images of the specimens into our database. So you can go in and not only see the data for the specimen, when and where it was collected and so forth, whether it's an adult or a juvenile or that kind of thing, but can actually get some images of the specimen itself. And then there are a lot of natural history museums that are, and we've sent a number of our specimens off in recent years for this purpose, but there's a big effort also to digitize the specimens themselves. So not just take a photograph of them, but to for example, just take it 3D scan the specimen. And, there's some real challenges in hosting that amount of data and so forth online. But what's really nice about that is in some cases, all the information a researcher might need from that specimen could be gleaned just from that, looking at those online images, particularly if there's a really, really nice 3D scan where the specimen can be rotated around and that kind of thing. And so then that would, preclude having to pack up and ship the specimen or from hosting a visitor to spend time working in the collection. But it's a very slow and fairly costly thing. But I suspect as we go forward, that's going to be a really transformative for natural history museum collections, is to be able to really image well their specimens to make those image data more accessible.
Very cool. We had a viewer wondering if you still do any hand logging of new specimens, or everything is straight to digital now, I assume.
That's a very good question. That's really very insightful. So the answer is we still do hand log. Most museums have pretty much stopped doing hand log, but what they'll do is they'll print out hard copies from the digital database as sort of a backup kind of thing. But we still do hand log first. And that's just simply because of precedence. It's just sort of, it's an old habit that's been going on for so many years. But we are unique in that sense. Most museum collections have sort of now bypassing that step, but we still do have the handwritten ledgers. But the database is great because the database, in addition to capturing all the information that's in the handwritten ledger, it really gives you a lot of flexibility to add in additional notes, remarks, comments, a lot more space to do that. So definitely digitizing the data are the way to go. So,
yeah, we had someone asking. Well, first of all, they said thank you for the great talk, and they wanted to know what species continue to fascinate you after many years of experience in the field.
That's a great question. Thank you. I'm usually just fascinated by the ones that I'm currently working on. So, the process of understanding what a species is and what are the criteria that we need to recognize it can be so complicated. And we just so often find ourselves in these really complicated situations where we might have conflict among data sets. So the morphology, for example, the body size or number of scales or coloration might sort of provide a line of evidence that these populations we sample belong to two different species. Whereas the molecular data, like the DNA sequence data, might tell something very different. Maybe they say, oh no, they're all just one. That's just one evolutionary lineage that we should call a species. And the challenge of trying to figure out what to merge these different kinds of data sets to make those kinds of decisions about what we want to call one species and what we want to call another, it's so complicated. And so what I'm always excited about is we're just constantly getting new tools. It's getting easier and easier to sequence data, DNA data, and easier to analyze large amounts of DNA sequence data. And so what I'm just really interested in right now is being able to apply some of these new tools to older projects, things where using technology that was not so advanced a number of years ago, revisiting those and seeing how some of those problems hold up given the new tools we have today. But honestly, my answer is it's really kind of what you're working on, what I'm working on right now. I get so engrossed in understanding this morphological and genetic, in the case of frogs, acoustic, the call variation, and looking at all the old literature and type specimens and so forth, and trying to piece together a puzzle that it's really kind of the species that I'm working on at the present moment is the one at the top of my mind. But there's an infinite number of projects to be done just in our collection alone. Many lifetimes of research.
Yeah, it's a really, really vast amount of information. We could probably talk to you for hours. Like you said earlier, you could tell stories for weeks. I guess everyone wants to know, when will the in-person tours return? Do you know yet?
In-person tours. So what I would say is, if you're interested in in-person tours, to just drop me an email and I'll hold on to it, and we can remain in touch for when things improve. And I'm optimistic. I'm much more optimistic today than I was this time last year at Reptile Amphibian Day last year, when things were still very, very, very unclear. But I'm really optimistic that we're getting beyond COVID, and we'll be able to do those kinds of tours. But in any case, they do require advanced scheduling. Even that building is closed to the public, and you wouldn't want to just show up for a number of reasons. So I would say, if you're really interested in having an in-person tour, just drop me an email and we can work something out when it's appropriate to do so.
Excellent. So don't show up to the address for a tour, and probably don't show up to the building with a specimen to turn in. How would the public submit a specimen if they thought they had something notable?
Best thing would be to contact us in advance, and so we can learn what it is and what might be done with it.
All right, and we've put your contact info in the chat for anyone. I'd like to thank you so much, Dr. Stuart, for sharing this with us today. Thank all of you who took your lunch break to join us, and for all the great questions. Please be sure to check out our other Reptile and Amphibian Day programs, which are going to be happening throughout March. And starting with Old News tomorrow, it'll be live at 1 p.m. If you want to check those links out, we'll put them in the chat. And we will see all of you soon, hopefully.
Thank you.